Hey, good morning. I'm a faculty at New York Medical College. It is a great pleasure to share our research product. Our research product is for food allergy treatment. It is in the category of botanical medicine. Here's my disclosure. In addition to my academic effort, I'm a co-founder of General Nutraceutical Technology with the goal to translate laboratory discoveries to the real world to help people with food allergy sooner. So I have a sub-award. I'm a sub-award PI of NIH Small Business Innovative Grant with GNT and some patent. I also have a compensation from the practice. Our botanical products, including the followings. One named FAHF-2. This is a herbal formulation that containing 9 herbs. We have an IND completed 2 phase 1 and 1 phase 2 trials. We further refined FAHF-2, developed the enhanced FAHF-2 product under the same IND as a sub-project. We have an ongoing phase 2 trial. From there, we further developed a botanical medicine with active compounds. We are planning to apply the IND as an amendment. The indication, as I indicated, that these botanical products are for food allergy. I will divide my presentation into a few parts. First is to give you an overview about allergy, particularly food allergy. Allergy is a global pandemic of a non-communicable inflammatory disease. According to World Allergy Organization, it estimated that 40% of the world population is affected by allergic diseases in a different clinical expression. You can see the environmental allergies, skin such as eczema, airways, digestive system like food allergy. Current approach include avoid the triggers using corticosteroids and other medications such as antihistamine and allergen immunotherapy. But overall, there's no cure for these conditions. Food allergy affects 30 million Americans with an estimated annual cost of $25 billion. Even with best practice avoidance, each year more than 200,000 ER visits, including children. As far, the treatment is very limited. Only one FDA-approved treatment, Parfazia, for peanut allergy. This is a powder of a peanut. This product was approved in 2020. However, this product itself sometimes also trigger reactions or even anaphylaxis. Therefore, currently avoidance and the use of rescue medication for anaphylaxis following accidental exposure is still the main management. Mainly once you have accidental exposure, you use antihistamine and observe the symptoms. If a symptom progress to severe, you will use self-injectable epinephrine and will follow up in ER. So the good news is that there are several interventions underway, including allergen-specific immunotherapy, different versions, and anti-IgE therapy. as well as Chinese medicine. As Steve mentioned that in the healthcare system, there is still a strong disparity. These biologicals sometimes very effective, but the cost is very, very high. Could be $20,000-$30,000 that is not practical for the people who are not insured. So therefore, herbal medicine, because giving the cost effectiveness, would be a potential for new product treatment for food allergy population. As you can see here, food allergy has been increasing dramatically in the past decades, from 4.6% up to 22% in 2016 from 2007. Food allergy can cover varieties, but there are several foods account for 90% of food allergy reactions. In 2004, to facilitate prevention, FDA requested food labelings on the 8 foods you can see here: peanut milk, shellfish, and soy. In 2021, FDA added another food, sesame, required a food industry to label this product. This is important to help patients with the food allergy to have a better prevention, but it's very difficult because some foods you can see as a shellfish, they have lots of different versions, tree nuts and the fish, and all of this, they have within this category, they have cross reactions. So therefore, food allergy has a very negative impact on people's daily life. What do we know about food allergy mechanisms? In general, from an immunological perspective, we have T helper 1 and the T helper 2. This is a general simplified theory. And the normal situation, Th1 cells help us to help our immune defense system against viral infection by producing interferon gamma. Whereas Th2 cells produce Th2 cytokines, help our defense mechanism against the parasite infection. However, if this immune system lost the balance, if we have too much Th1, we may develop autoimmune disease. On the other hand, if we have too much Th2, we'll have allergy. This illustration tells us why overexpression Th2 will cause allergy. Taking peanut as an example, in a susceptible individual, when they ingest the peanut, they will produce more Th2. These Th2 cells promote B cells to produce an antibody. This antibody called IgE. I named E as evil because this antibody can bind to mast cells. These cells has pre-made histamine. Once the individual re-exposure to the same allergen will trigger histamine release, you know histamine causes clinical symptoms. What's the symptoms of food allergy? There are different stage and different symptoms. You see hives, could be mild or very severe, or edema. You see this situation, it's very dangerous because the throat might be closed too. And then you can also see vomiting, GI symptoms, and progress to respiratory symptoms. Both situations can directly progress the condition to a very difficult situation. Patient could have lost consciousness and in a very critical condition. At this stage, it is life-threatening anaphylaxis. In the second part, I would like to discuss the development of IND-FAHF-2 and the EB-FAHF-2 from traditional Chinese medicine, TCM, for food allergy. We adapted a TCM anti-parasitic formula called Wu-Mei-1 and developed the food allergy formula 2 that contains 9 individual herbs. As you know, parasitic infections cause a Th2 response. This response drives elevation of IgE, activation of mast cells. You can see they share the key mechanisms with food allergy. So this is our principle to initiate our research. We use an animal model. We showed that FAHF-2 exhibited excellent safety and a very strong reproducible efficacy to reduce or prevent anaphylaxis in model. I will show you some of the slides in a few minutes. So given this efficacy and the traditional use, FAHF-2 received the FDA Botanical Investigational New Drug for food allergy. This is the first botanical product receive IND for food allergy. So we conducted the phase 1 and 2 trials to show the excellent safety as seen in animal model and the immunomodulatory effects. We also developed a refined version of FAHF-2 named the EB-FAHF-2 in phase 2 trial. This refined product is still under the same category of IND. In this trial, we combined with a pharmaceutical drug, anti-IgE, and currently widely practiced the treatment oral immunotherapy. Some of our publications listed here, but for today, I will focus on the studies in the following 3 aspects. This formulation received a US patent. This is a 3-dimensional HPLC fingerprint of FAHF-2. There are 2 key herbs, philodendron and Ganoderma. This is a HPLC fingerprint of FAHF-2. As we discussed before, the herbal medicine is very complicated, but you still have a way to identify the key compounds for the product quality control purpose. This is the animal model we used to evaluate the effect of FAHF-2 on anaphylaxis. This is normal mice. Before challenge, this one was sensitized with peanut. Before challenge, you see everything's normal. However, 30 minutes after challenge, this individual developed anaphylaxis. You can see puffy eyes, nose, and purple color means lack of oxygen. So we give this score as 3. Here is a summarized data showing FAHF-2 has persistent protection against the peanut anaphylaxis. Just give a little bit of detail here. This column shows the time after challenge. We challenge it from week 15, first challenge, and all the way to 7 challenge, because this study want to show the persistent protection. This is peanut allergic mice, PNA, received a sham treatment. In this study, we give water. You can see the reactions 13 out of 13 with a score 2 to 4 4 is near fatal reactions, 5 is fatal, 0 is nothing. And then you see that this group of mice developed a near fatal reactions. Actually, one individual did not make it. However, when the peanut allergic mice received the FAHF-2 formulation treatment, you see it, we see a full protection all the way to week 40 until week 70, there are some effect decline. The normal mice has no reactions. We then look at the immunological mechanisms, focus on IgE and the IgG. IgE causes reaction, IgG is protective. So we see that here in FAHF-2 treated mice, showing in this open line, had a reduction of IgE, but an increase IgG, suggesting an immune modulation on B cells. We also look at the T cells on Th2 and the Th1. We see reduction of Th2 and the increase of Th1. And then we look at the effect on effector cells, mast cells, as I mentioned, that release histamine cause clinical reactions. We see that in sham treated mice, you see lots of granules that containing histamine release, where FAHF-2 mast cells show the very good integrity, almost like a normal control. So this summarized data, you can see the significant reduction of mast cell activation. Now I'm going to show you some slides about the clinical observation. This is the acute phase 1. This summarized the data. This is a 1-week study. This is a double-blind placebo-controlled dose escalation study. The FAHF-2 group has 12 and placebo group has 16. You can see everything all in the normal range. In this study, we look at IL-5 as a marker of Th2. We see that in FAHF-2 treated group, there is a significant reduction, but not a placebo group. Just to let you know that unlike animal study, the data are very universal, consistent. But in human, you can see that the individual results have a very diversity. So we also conduct a 6-month safety study, but same as this is an open label study. We found consistent safety. You see everything on the liver, kidney function, glucose, all in normal range. In this study, we also look at the effect on basophils. Basophil is circulating cells, similar like mast cells. They release histamine. In the clinical study, we use peripheral blood to look at the basophils. We see after 6 months, there is a significant reduction on basophil activation. Although FAHF-2 has demonstrated to be safe and have a good immunomodulatory effect, the challenge is that traditional formulation has a very, very big daily dose. So when we convert the decoction to the capsules, patients have to take, the full dose will be 36 pills. So that affect the compliance of a patient to keep up the dose. So therefore, our research has been focusing on how to refine this formula to reduce the daily dose while remain the efficacy. We found that use butanol or ethyl acetate purification. This is the safe solvent normally to purify the to reduce the caffeine from tea or coffee. So we use this method, the FDA approved method. We were able to purify FAHF-2. We are able to reduce nearly 80% of daily dose while maintain the same effect. We have published this data. With this observation, we received the FDA's approval to transition from FAHF-2 to EB-FAHF-2, and then we started a new trial. This is the trial protocol. This is a combined trial, as I mentioned that. This is the FAHF EB-FAHF-2 together with omalizumab anti-IgE, with the goal to reduce the reactions. And then we started oral peanut immunotherapy. Here has two groups, EB-FAHF-2 or placebo. We completed most of the trials and now in the data analysis. To give a quick summary on this FAHF-2 and the EB-FAHF-2 development, FAHF-2 is the first US FDA botanical IND derived from a TCM antiparasitic formula. It demonstrated a high safety, complete protection, and immunomodulatory effect in food allergy clinical trials. Reproduce the safety, immunomodulatory effect, clinical protection remained to be established. So refined FAHF-2 named the EB-FAHF-2 received the FDA approval for phase 2 study as a sub-program of FAHF-2. Clinical trial is ongoing. So the part 3, I want to share with you our newer development. While we are continue to develop the formulation for treatment of food allergy, with the technology, we also began to understand the active compounds. And we found that from FAHF-2 formulation, one compound that seems like important, because we did all screenings from individual herb to fractions. We found that this compound has a potential in the cell line. We then look at the anti-IgE effect in human peripheral blood mononuclear cells. So we see a very strong suppression. You see at a very low dose, berberine almost eliminate the IgE production. We have a patent received just recently. Although berberine showed a strong effect in vitro, berberine has a poor bioavailability. It did not show effect in vivo. To increase the viability, we adapted nanotechnology to increase the presence of compound in the circulation. As you can see here, this is the concentration of berberine in the nano encapsulated product. We see a significant increase of bioavailability. In liver, we see nearly 10 times a higher concentration of active compounds. We conducted a safety, showed an excellent safety profile. We fed mice 14 times daily dose and did not see any abnormality. We next look at the efficacy. This is the protocol we used. First, we sensitized mice with peanut, and then we treated them with different groups. We left untreated with the compound alone, with the nano product, with Mark Nano, and left untreated. Importantly, after we finished the treatment, we left the mice for many weeks up to week 28, and every 4 or 6 weeks we challenged them to see the persistent effect. After the final challenge, we evaluate the effect by looking at the IgE, look at the anaphylaxis score, including body temperature, plasma histamine, and collect the T cell, B cell, and then we have established the MOA. But for today, I will just show some of the slides. Here is the effect on IgE. As I mentioned that IgE play a central pathological role in peanut allergy. If we are able to suppress IgE, inhibit IgE, the therapies have a good potential for treating food allergy. In this figure, we pre-sensitize mice. This is the pre-therapy. We make mice allergic to peanut, and then we start a treatment. Just give you orientation. The red line indicating the sham, peanut allergic mice. The blue line is the compound alone, and then this line indicate the mark nano. The most important one is this orange one. This is nano encapsulated compound. You can see that as early as 4 weeks of treatment, IgE had a very sharp reduction, mostly interesting even after we stop the treatment. Here we complete the treatment, IgE continue to reduce and approach to baseline. So this data indicate a very persistent beneficial effect. Here I want to share with you the effect on anaphylaxis protection. This is the symptoms score. 0 is nothing, 5 is fatal, 4 is near fatal, 3 is respiratory symptoms, 2 is a GI, 1 is skin. So this is the first time challenge after the treatment. You compare with the sham, 100% of mice develop anaphylaxis as well as compound alone without a nano capture or mark nano. The compound with nano encapsulation showed over 90% protection, only 1 showed a mild reaction. Interestingly, when we continue to challenge mice, we see that later on, they actually fully protected, even the therapy stopped. So now I'll give the summary about this new development. Berberine, as you know, it has been commonly used as a dietary supplement, have a lot of health benefit. But before our discovery, no one had reported that berberine has effect on IgE production. So therefore, our discovery have received the patent approval issued. The developing a clinical drug required the nanocapsulation of berberine to prevent degradation and enhance the GI tract absorption, because berberine, as I said, has a poor bioavailability. These problems blocks the oral application. Therefore, nanoencapsulation of natural compound has a good potential. This product also showed a striking effect on bioavailability and the reduction IgE. As I mentioned, that continue the protection post-therapy is also very unique and most impressive. So now here talking about a little bit of market opportunity. So I particularly thank the organizer to provide this opportunity to share this information. So food allergy has been rapidly increasing in the past 2 decades, affecting 32 million Americans with annual cost of $25 billion in the US. At the present, there is no cure. Treatment is very limited. As I said, only 1 product for peanut allergy has been approved. The Pavazia was developed by this company, received the FDA approval in 2022. It's very new, and then later on, this company was acquired by a French company with this much value. So our plan is that we are completing the preclinical data to prepare for FDA amendment to include this as a sub-project. In part 1, we want to further generate CMC and PK data, further develop a toxicology data, pharmaceutical, and finalize a clinical trial. Then the part 2 will file IND as amendment to a current IND with the goal to receive IND and IRB application. This will take 1 or 1.5 years. We will then conduct a phase 1A trial, which include 40 patients. This will be a dose escalation study that needs 6 months. Then we will move to 1B to study PK. Because this is a single compound, I think it would be easier to study PK. And then we will move to phase 2A, focus on the biomarker study, because food allergy is quite dangerous. We do not want to put the patient at a risk. We will use a biomarker to indicate that the therapeutic effect. After we complete this phase, we'll move to a double-blind, placebo-controlled oral challenge phase with a goal, of course, to move to the NDA phase. With that, I want to acknowledge my research mentor, Dr. Hugh Sampson. He has a vision and a support for traditional Chinese medicine, for a new drug development for food allergy, and my colleagues, collaborators here, and the clinical collaborators and the families. Want to thank them for their inspiration and support and participating in the trials. Here is the funding agency. I want to thank NIH, private organization, foundations, individuals, and small business grant support. Thank you all for your attention.
Thank you, Dr. Li. And I know you have been a pioneer in the botanical drug herbal medicine development. So a long story short, how long have you devoted your time in developing this product? What triggered you doing that?
Perhaps, yeah, proportionally lots of time to this development. But fortunately, you know, we have a team. For the botanical part, we have a Dr. Yang lead the team. For the clinical trial, we have a Dr. Sampson and my colleague, Dr. Julie Wang. So, and because this team has been established in the food allergy clinical and basic research, we were able to have other research institutions like Hopkins, Little Rock, Arkansas, and other university. Therefore, the trials, you know, it's in the good pace because a lot of institutions involved in the trial. But still, we are still facing lots of challenge. We would like to work with you and Steve, you know, to accelerate this project to make it available for all patients, you know, for all patients, regardless the background. Yeah.
Great, yes. David, Steve, and any others have any questions?
No, excellent presentation, Dr. Li. Good to see you again. David Allen, I'm in San Diego.
Yeah, okay.
Yes, yes. It's good that you've done the nanotechnology encapsulation now. So how many capsules are you left with now with the nanotech?
For nano, now we were even better. We use a syrup.
Oh,
we
that's
not use
fantastic.
capsule. Right.
That's brilliant. That's wonderful.
Yeah,
I'll reach out to you again. That's fantastic.
Okay. All right. Take care. Thank you. So good to see you.
Excellent work for a very, very important topic. Great presentation. Excuse me. I also appreciate the fact that you can make the argument once again about the price, the income, in order to make the drug affordable and accessible. Yeah, this is very interesting. Important topic, very important issue. Have you gotten any food company sponsors in terms of your funding?
Well, that's a good question. We had some connections, but have not make a deal yet. So yeah, we continue to open opportunities. Certainly, even we have some finalized collaboration, we'll still need a strong funding. We can finish the phase 3. For phase 3, I plan to have 10-20 sites, right, with our capacity collaboration.
Of course.
Yeah, so that we'll have more participants, for people with all background.
Indeed. And have you been approached by pharmaceutical companies? Early on, yes, but I think now the pharmaceutical company, most of the time they want to see you finish the phase 2 trials. Correct. So correct.
The early stage trials is the most difficult. What we rely on NIH grant, slow, right? But we have something to support to continue, and the families, the private donation is
indeed
it's a major support, right?
Very important.
Yeah.
Indeed. Excellent work.
Yeah.
I look forward to working with you.
Yeah. Now we need you, Dr. Low. Everybody to be able to as a team to support. Yeah.
Yeah, we will all work together to face the challenge of we're not being paid enough attention in the drug development yet, but we are raise the public awareness, and so that more people realize developing herbal medicine to prescription drugs may be an efficient drug development pathway.
Right. So, thank you. Yeah, it's a challenge, but need just the persistency continue to make the effort.
Sure.
Yeah.
Okay,
thank you.
There's a question from let's say, food allergy may need local acting varying. Please explain why nanomedicine is used, which increased bioavailability in systematic circulation.
Say that again.
Okay, in the chat, there's a question to you.
In the chat, okay.
Yeah. Severo, do you want to explain your questions to Dr. Li? But I'm going to read the question.
Okay.
He said, food allergy.
I see it. Now I see it. I just turned off my slide. Locally, food allergy may need locally acting berberine. Because please explain question. This is an excellent question. I think to explain this is that the immune cells, particular IgE producing cells, mainly in the spleen, lymph nodes, and the bone marrow. So the active compounds need to be able to circulate to that organ area in order to play an immunomodulatory effect, or in another word, reduce IgE. But this is an excellent question. However, berberine can treat diarrhea because the bacteria is in the GI tract. That does not need any nano product to enhance the bioavailability. But for the systemic problem, the bioavailability is important. I hope I addressed the question.
Great. I hope that answers your questions, Lino. And great. And with the time, and thank you again, Dr. Li, for the great presentation. And we'll follow up on this product discussion at a later time. Thank you for your time.
Thank you. Thank you.